Dr. Saeed Al Turki is a consultant of molecular genetics, genomics, and bioinformatics, and the head of the translational and molecular pathology lab in Ministry of National Guard. And as usual, we know Dr. Saeed just show us a very interesting presentations, and we expect to enjoy this lecture. Please, Dr. Saeed.
Thank you very much, Dr. Ahmed, for the kind words. Assalamu alaikum wa rahmatullah. Good morning, everyone. And thanks for the organizing committee for their kind invitation to speak about the role of molecular pathology in breast cancer in general, not only in HER2 positive. And in preparing for this talk, I was thinking about the axioms or the perspectives from different stakeholders who's working in the healthcare of breast care management. And as you can see, there's at least 4 different axioms or perspectives. It means different things for different stakeholders. In clinical needs, a histopathologist may need to know the molecular classification of the breast cancer, while the oncologists want to know the predictive and prognostic value. When it's come to the inherited risks, we need to differentiate the somatic versus germline. Germline is becoming more and more very important. Up to 15% of the patients have BRCA or germline mutation. 5% of them would be actionable. The 4th axiom is the specimen availability. So tissue is always the golden standard, but more and more we are using blood as well as the liquid biopsies, whether it's cell-free DNA or the circulating tumor cell. And circulating tumor cell recently showed a poor prognostic value. Just the number of CTC in the blood itself have a prognostic value, although it's not part of the guideline yet, but it seems to be a very hot topic in the next couple of years. And of course, from a lab perspective and hospital, cost and infrastructure is extremely important. These tests are very expensive, and whether we go for single gene testing or for panel testing has a huge impact on the infrastructure. So to understand these axioms, I will share with you 2 different experiences recently published, one from Japan and the other from Italy. In the Japanese experience, which was published a couple of months ago, they used Foundation Medicine tests, and they are comparing it also to the standard of care, the immunohistochemistry for HER2 or the FISH, plus comparing it with the germline testing, the BRCA analysis from Myriad. As you all know, this test was FDA approved in May 2019. And in the context of HER2, it was approved for the ERPP2 HER2 amplification, as well as the PIK3CA mutations. But it also comes with the value of adding tumor mutation burden and microsatellite instability for the immunotherapy. If you map these axioms to this test, we will see this test will help for the prediction of the therapy selection. It also enables us to understand the somatic as well as the germline mutation. It's based on tissue, and there is a version of it on the liquid biopsy. And it's considered a panel testing. It's a send out, expensive, north to $2,000 per patient, up to $4,000. So this paper selected 120 cases. Almost 109 of them were sequenced successfully, which is 95%. That's very good. And the average, the mean age was 62. The subtype was laminal P in 40%, followed by triple negative, also in 30%. And more than half were stage 4. The frequency, so there's 2 major groups of mutation that we can pick up from this assay. One is the single point mutation and small insertion and deletion. And the interesting thing here is almost 108 patients out of 109 were positive for at least 1 mutation, which basically means almost 99% will have actionable mutation. Now, actionable means different things. In 50% almost was TB53, so it's not that interesting. But it's followed by PIK3CA in 40%, and you can see number 3, 4, and 5, which is GATA3, BTN, and BRCA1 was 11% in each. If we look only in the hormone receptor positive, it's BIC3CA was the most commonly frequented mutated gene. And in the HER2 positive, TB53 was the first one, and similar story in the triple negative. So let's go to the other type of genomic alteration. In this case, the losses, the amplification, the gene fusions. And you can see the most commonly mutated is the ERPB2, HER2, followed by MYC and RAD21. In the hormonal receptor positive, RAD21 and MYC was the mostly common mutated in 20% each. HER2, unsurprisingly, ERBB2 was the most important and commonly mutated. And in the triple negative, it's a completely different story. So mostly what we see is the orange color, which is losses. So BTN, CDKNA, and MTAP. And if we summarize all of this data in one graph, you will notice that almost 42% are amplification, followed by missense and frame shift. And there is a very important message here. Because more and more we are using next generation sequencing picked by NGS, except in this test because it's used capture hybridization technology, which is very good for amplification. But most hospitals are actually using amplicon based, which is not good at all for amplification. So by using NGS in-house, if it is amplicon based, you need to be worried about the amplification and use the alternative test, whether it's FISH or send for foundation medicine. And if we go for the actionability, this diagram shows two different things. The inner circle is the breakdown of the mutation. So ERPB2 is 25%, BIC3CA is 18%. And the outer circle, which is the green color, is the level of evidence. So that's level 1A. So from this data, we can pick up to 50% or 45% of our patients with a level 1 actionability using only foundation medicine, followed by BRCA mutations. Another good question was, how can we compare the foundation medicine with the standard of care in HER2, the immunohistochemistry and the FISH? And the good sign here is the concordance was almost 96%. But again, this is only the hybridization, not the amplicon-based, and further work needs to be done to evaluate the in-house methods. When it comes to the microsatellites, only one patient showed a very high microsatellite instability, and it turned out to be a Lynch syndrome. Tumor mutation burden was picked as a high in 3 patients, which is almost 3%. And it was found that it's usually high in triple negative and laminal P, the high tumor mutation burden. Now, another thing the authors did is they compared the BRCA germline. They sent it previously to Myriad, and they tried to see how's the correlation between Myriad test and the tissue-based somatic in foundation medicine. And let's see this green box at the top. All the germline mutation here was picked by both, from the blood and the tissue. But the second half, which was negative from Myriad tests, was positive in the foundation medicine test. And most of these are somatic. So, which tells us that by testing the tissue, you can pick almost all the germline mutation plus doubling the number of BRCA mutation from somatic. Which doesn't say that BRCA analysis from Myriad is bad, but it's the sample tissue, the sample type. From the blood, we cannot pick all the somatic mutation, but it's true the other way. So the second experience from Italy, and it's a review mostly about the oncotype and the multi-gene breast cancer classification, oncotype DX, EndoPredict, MammoPrint, and Prosigna. These are the most important 4 tests available today for molecular classification. And basically, all of these looking for gene expression using different methods, and they will output a report for the recurrence and the molecular classification. But when we look at the details of these tests, which most of them are sent out, so it's not in-house. Oncotype DX, they use 21 gene, and the technology they use is BCR. Mammoprint uses up to 80 genes, and they use next-generation sequencing. Followed by EndoPredict, 12 genes, BCR, and the Prosigna is 58 genes, and it's a direct hybridization. What does that mean? If we plot the technology behind these 4 tests, you will find that Oncotype and EndoPredict use BCR, which is very sensitive, but it's not highly multiplex, so only few genes can be tested. If you look at the Mammoprint, it uses next-generation sequencing, so it's extremely huge number of genes if you want to, but the sensitivity is not that great compared to the qPCR. The 4th one, which is the Prosigna, is based on nanostring, a new technology that's worth actually explanation. And it's as sensitive as the BCR and a little bit more when it's come to multiplexing, but it's not as big as the NGS. So this is promising technology, has a very clever methodology behind it. So it goes for every transcript and it's tagged with multiple colors. And you can see here, the red, green, blue, red, green, yellow. This sequence is very unique to one gene only. So it will attach to it, and it's like a flag or a flag that will be big as a proxy to count the number of the gene expression in that gene. And once we do this and we tag with different sequence of colors, they will attach into a surface. We apply electricity, and then a laser will go and scan it in these positions, and it will read the sequence of these colors. And the end result is this. It will count P53 as 1, EGFR as 2, because everyone has a very color code specific to it. Why this is important? It's important because classically in genetics or molecular biology labs, we always go from RNA and convert it to complementary DNA first. And when we do that, you see here 3 color of gene. There are 3 genes here, green, orange, and blue, and we have 3 copies of each. Once we convert, we lose some of those transcripts. And you can see the green gene is only 1 copy. It used to be 3. And then we have to amplify to increase the signal. But because it's already lost, there will be a variation in the copy that does not reflect actually what's happening in the patient. So in the real patient, in the real sample, it used to be 3 copies. But eventually we'll have a differential expression, and this could actually ruin the prediction algorithm. Nanostring does not do the amplification, does not do the conversion. It's actually attached to the RNA directly, and it's have an absolute count, which make it extremely good at predicting the reality in the tissue itself. So what is the output of these tests? One, it will tell us about the therapy, the prediction of use of chemotherapy, as you know. Some of them will give us a risk of recurrence within 10 years. What's that risk? And 2 of them will give us a molecular classification to the 4 types, laminal A, laminal P, HER2 enriched, and basal-like. So the question now, are these results from these tests interchangeable? Can we use it to predict each other? And the answer, unfortunately, is no, for multiple reasons. Different technologies, different risk, continuous calculation, and there is at least 30 different studies that compare each other. There is no correlation between them. Is that bad or good? Let's see. This table will summarize the evidence from ESMO, AGCC, ASCO, and NCCN. And it seems like Oncotype, it's the oldest one, and there's multiple plethora of trials on this. Evidence 1A in the ESMO, it's a level 1 in AGCC. It's very strong in the ASCO, NCCN guidelines, endorse it for predictive and prognostic, and it's the level of evidence is 1. So it seems to be the most preferred method in this context. If you look at the Prosigna, it's level 1A in the ESMO, but level 2 in the AGCC, and it has a strong evidence in the ASCO. And NCCN guidelines says it has a prognostic, but not predictive value. But doesn't mean it cannot do that. It just needs some work to be done. And it's the youngest of all the technologies. I won't go into the EndoPredict and the mamma print, but they have a medium or in the middle, basically, when it comes to the quality of evidence. But nonetheless, there is 38 trials, a meta-analysis done on them in the ASCO in 2018, and compare the clinical utility, and all of them have some value. You see here the chemotherapy use percentage is only 31% in the oncotype compared to 49 in Prosigna and above 50% in the others. There is the 10 years total cost of care saving is also comparable between them. But the idea here and the message is they are not interchangeable, but they are useful in each in their own. So how can we, before using one of these tests, there's 3 questions that we need to ask. First, in the question in the red, is there a need for genomic expression profiling? And the answer is, if the patients have a positive hormone receptor, HER negative, early breast cancer, then go ahead. Which of the available tests should be performed? Only one. Please don't send ever for 2 because it's not correlated and you might end up having conflicting results that will confuse you and confuse the patient. And ASCO and ECCN is not endorsing one over the other, but they are bringing the evidence. So we will wait in the next couple of years to see how this pan out. And how should the test results be not going to undergo chemotherapy, either because of the clinical status or they're refusing it. And if the clinical pathological features are more instructive. So finally, just a very quick talk about the liquid biopsy rule. As I mentioned, cell-free DNA is becoming used more and more, especially for BIC3CA mutations from the blood. What's inside it, just counting it, will give us prognostic value, usually associated with poor prognosis, but it's not yet part of the guideline. And I'm ending with this slide from the latest version of NCCN guidelines, which show how molecular pathology rule is getting more and more actually in all aspects, whether it's germline or band cancer like the NTRK or the immunotherapies, MSIs, and tumor mutation burden. So the role is getting more complex, which is good news for the patient specifically by giving them more options. And
thank you very much, Saeed, for excellent talk as usual.